Nani record. Okay, very good morning, my dear friends. So today's our eminent resource person, Dr.A.Arajani Madam Garu, Assistant Professor, Department of Botany, Government Degree, Government RBVRR Women's College, Autonomous, Hyderabad. Madam, please welcome. Madam has explained yesterday about the edogonium, has clearly explained about the edogonium of the chlorophyce member. So regarding of that class, we hope clearly you are already listened. But today,
Yes, sir.
Madam will going to explain about the one of the important type study of the chlorophyce member, Chara. Madam, please share your screen and start your session. Madam, please welcome.
Okay, sir. Thank you. Sir, visible my slides? My slides are visible?
Madam, yes, your slide is visible.
Okay.
Please
Okay.
click on slideshow. If possible, please click on slideshow.
Yes. Here option is not there. One second, sir.
Animation పక్కన ఉంది.
Wait. Open అట్లే సార్.
Animation పక్కన పైన ఉంది చూడు.
అవును.
Animations పక్కన రివ్యూ.
అది క్లిక్ చేస్తే ఓపెన్ అవ్వట్లేదు.
వస్తది. స్లైడ్ షో క్లిక్ చేయండి వస్తది. లేకపోతే ఈ పైన ఉంది చూడండి, ఆ పైన ఉన్నది ఆ లెఫ్ట్ ఇంకొంచెం
లేదు స్టార్ట్ అయింది. లేదు సార్ స్టార్ట్ అవుతుంది. 1 second. Yeah.
Right.
Now it's okay.
Okay, right.
Yes. Good morning, students. Good morning.
Yeah, ma'am. Good morning.
See, in our topics are going on algae, no? Algae general characters, reproduction, classification, and the chlorophyce members also. I'll start the chlorophyce members. That one is the first class volvox. Yesterday class is the edogonia, and now today we will
Chara.
study the Chara. The type study is Chara. Here, this is the macroorganic. Yesterday, 2 classes are there, no, in volvox and edogonium. Both are the microscopic. You can observe to the under electron microscope only. But the Chara is macroscopic algae. This one is the macroscopic algae. See here, Chara, the classification. This is also chlorophyce member. The third type study is called in degree standard chlorophyce member. Last type theory also, this one. This is the Chara. The class, you know, chlorophyce, the order Characeae, family Characeae. Another name is called the Chara. This is the classification. Most probably not ask the classification when you are write the theory part. In that time is important. Okay, leave it that one. What is the importance of Chara? What questions are to expect the Chara? Here you can take the habitat of Chara. You know, all are the freshwater algae and chlorophyce. Here the plant of Chara, you know, that one is the macroscopic algae, highly developed, filamentous, branch like higher plants, differentiation into the nodes and internodes, laterals, internode laterals, rhizoids also present. And your algae grows abundantly in freshwater, Next one is a still and shallow water reservoirs, attachment submerged aquatic algae, muddy, sandy, bottom of pools, ponds, lakes, ditches, like this. Okay. Okay, coming to the another important character, these Chara. The Chara, you know, the algae cell wall to made up of cellulose. That is a very common. But here in this Chara, the cell wall or plant body to made up of the calcium carbonates. These are the two expected questions. Very important. The Chara plant body to made up of the calcium carbonate. That's why the Chara name is called stoneworts and brittlewort. Very important. This is the expected question. Chara plant body made up of to the stone or calcium carbonate. The most important characteristic feature of the Chara is the deposition of calcium carbonate all over the plant body. That's why due to which is the body surface become very rough, quite rough. It can be brittle to touch. When you can touch the Chara plant, brittle to touch and very rough. It can be released touching the body. This is called, that's way the plant body name is called stoneworts or brittleworts. Very important this question. What is the way you can the plant body studied the in algae? In the Chara, the plant body studied the stonewort. The plant body is called the made up of calcium carbonate. That's why this is the stoneworts or brittlewort. These are the example. Vulgaris, Gillankia and Nuda. These three are the best example for the Chara. See, this is the occurrence, you know, Chara freshwater, green algae, submerged, shallow water, ponds, lakes, ditches. These are the very common. And growing in the brackish water also, hot water springs also you can observe. And Chara mostly hard freshwater rich in organic matter. In where the Chara is there, rich in organic matter, calcium and deficient in oxygen. This is also important. These plants to made up of calcium carbonate, that's why they called as brittleworts or stoneworts. This is the this one is the onion-like odor. When the smell is there, emit discernible onion-like odor due to presence of sulfur components. The smell of algae also, this Chara also, that one is the onion smell. Why onion smell odor is there? In that one chemical compound sulfur also present. That way the smell is like that. In our India, 30 species are identified. Scientists see Gillankia, Branu, Gracilla, Hat. These are the our Indian species of Chara. See, next one very important character, thallus structure. These Chara, one second. Yeah. Thallus, you know, plant body name is called thallus. But compared to other two chlorophysin member, one is the Volvox Idogonium, microscopic structure. The Volvox plant body, Cenobium colony. The Idogonium plant body, filamentous unbranched. See the sequence of thallus organization. Volvox is different thallus. Idogonium also different thallus. Here, Chara plant body, same thallus, but you can observe to the nodes, internodes, branches, rhizoid-like structures, macroscopic structure. You can see directly. This one main submerged, you know this part. Main axis and rhizoids. Here branchings are there. Each and every branch to contain to the nodes and internodes. Node is the two central cell. 6 plus 20 cortical cells are present. Internode only one axial and one cortical cells are present. And this is the branches. See, next one is the rhizoid. See, 1 second. This is the Chara plant or Chara thallus. See, this is the main axis. Just like tree-like structure, no? but it is the visible macroscopic. This is the plant body or external feature of Chara. This is the main axis and branches are there. Each and every branch to contain nodes, and see this part is node, this part is node, this part in between nodes is called internodes. And see here, next part is called the branches of the rhizoids also present. These rhizoids to help to the attachment of any substratum, to attach the any substratum. See, this is a rhizoid. What type of rhizoids? In sometimes to question the rhizoids question also. Here, rhizoids are white color, thread-like structure, to made up of multicellular, unicellular, and branched type of rhizoids. Very, very important. Where the in the branched type of rhizoids present in chlorophyll member, that is Chara. Rhizoids is there, but white color, thread-like structure, multicellular, unicellular, and branched structures are there. Branched type of rhizoids. The rhizoidal arise from the rhizoidal plate, which form the base of the main axis. Where the main axis there, in that main axis base, the rhizoidal cells are to form. That is the peripheral cells to made up of. What is the characteristic feature of rhizoid? Obligate septa is present. Help in the attachment of plant to any substratum. That is mud or sand or absorption of minerals. These are the importance of rhizoids. Okay, in sometimes these rhizoids to help into the vegetative reproduction. The vegetative reproduction, multiplication of plant, forming bulbs, secondary protonema. In the vegetative reproduction time, I will explain. Here one very important questions are to expect the question. See, this is a rhizoids. Very simple, no? These are the important character, multicellular and branched rhizoid, oblique septa. These are the very important in this rhizoid part. Coming to the main axis, you know, main axis is there, branches are there. Yes or no? Each and every axis, main branch and branches, both are to made up of to the nodes and internodes. See, this is the main axis, erect, long, branched, differentiated into nodes and internode. See, this part is node, this part is node. In between nodes is called the internodes. These internode consist of a single elongate oblong cells. You can observe them. Internode, very single, oblong, and consist of elongate cells are there. These internodal cells, some species may be surrounded by a celled, thick layer celled cortex. See this one, thick layered cell. Each and every cell to surrounding cortex cell. Such species are corticate species. Some species which cortical layer is absent, in that one is acorticate. The internodal cells, some species corticated cells are present, that is the corticated species. When this internodal corticate cell is absent, that is the ecorticate species. This is the internode structure. Where the node part, this one is node, this one is node. Node consists of a pair of central cells, small cell, surrounded by 6 to 20 peripheral cells are formed. Each and every node to surrounding by the 6 to 20 peripheral cells. The central cells and peripheral cells arise from a single nodal initials. This is the main axis. See, this is the main axis. Main axis branches are the nodes and internodes. I'll explain. Next one is the branches. Here, two branches are present. One is the branches of unlimited growth. Second one, branches of limited growth. See, the main axis is there, no? This is called the branches of unlimited growth. Second one, side branches are there, no? These branches are all are the side branches is there. This is the branches of unlimited growth. Second one is the branches of, this part is called the branches of limited growth. First one is first you can take the branches of limited growth. the arise in a row of 6 to 20 peripheral cells of the node. Node to form the peripheral cell node, 6 to 20 peripheral cells of nodes, main axis, branches on unlimited growth. That is called the unlimited growth. These are called bracklet branches or primary laterals. See, these are the unlimited growth is there, no? That is part is called the primary laterals. These branches stop growth after forming of the 5 to 10 nodes, hence they are called branches of limited growth. Where the primary laterals are there, in that primary lateral to produce to the 5 to 15 nodes, when that is the limited growth. Second one, the stipulodes. See here, each and primary branches is there, no? Branches one haze-like structures are, that is the stipules, stipulodes. These stipulodes and reproductive structures are formed where the node of this branch, where the node sites are there, in that node part to produce to the sex organs or reproductive organs. This is the limited. Unlimited growth arise from the axis of branches. This one, unlimited growth is that the middle part is unlimited. The side one is the limited growth. The branches of unlimited, this part, the branches of unlimited growth arise from the axil of branches of limited growth. Hence it is called the axillary branches or long laterals. These are differentiated into the same nodes and internodes. Nodes bear primary lateral, bracklet-like structures are main axis, and these grow, their growth also unlimited growth. These are two branches are present. See, I'll explain those stipulodes. See, this one is the main branches. Okay, these are the primary branch. See, these are the secondary laterals, and this is a node, and in between internode, and these all are the unlimited growths. See, this you can observe to the this is the external morphology. This part is the shoot of limited growth or primary laterals. Here, where the node is there, no girls, see, students, this one node and this one is node. This node part to produce the sex organs. Then secondary laterals also to produce. Each and secondary laterals to form to the stipulodes. So see, these are the stipulodes. Here, main, this is the main axis, node part and stipulode. This one is the primary laterals also present. This is the secondary laterals. This one is the primary laterals. Then the basal node of the branches of limited growth to develop and short pointed single cells outgrowth, that is called the stipulodes. Chara species, Barmachia, many species are there. Then these stipulodes, each node is twice number, primary lateral. That species name is called bistipulate. In some species of Chara, Nuda, Chara, Bruni, the stipulodes, each node equal number of primary lates are present. That's the such species name is called unistipulate. When the stipulodes present in one row, each node, the species are called haplostephanous. Which row? This is the, in case the two rows are present, diplostephanous. One stipulode is there, haplo. Two stipulodes are there, that is the diplostephanous. And the coming to the cortex region. Many species, Chara example, Infera, Aspera, the internodal cells, main axis, ensheathed cortex cells. These cortex species, this is the corticated species. Cortex, vertical elongated narrow cells are present. Where the internode, this is a node, no? This part, the internode of its length by corticating filaments are present. Upper node, that is called lower internode. This one, there is the ascending and descending filaments are meet in the middle of the internodal part. This is called the ecorticated. Examples, Chara, corlina. And coming to the cell structure. See, here nodal cells is different, internodal cells is different. Nodal cells are very small in size, isodiametric. Dense cytoplasm, uninucleate, ellipsoidal chloroplast, and vacuole is absent in nodal part. Sometimes it was very small vacuoles also present. Cytoplasm differentiated into the exoplasm, endoplasm. This is a nodal. Internodal cells elongated in size. Cytoplasm same present. Central vacuoles are present. This is a uninucleate. This one is a multinucleate. Contain many discoid chloroplasts. See, here ellipsoidal chloroplast is there. Where the nodal part is there, discoid chloroplast, and differentiated into the exo and endoplasm. Now, coming to the here expected questions to ask the vegetative reproduction. In the vegetative reproduction, total 5: amylum starch, bulbils, amorphous bulbils, tubers, secondary protonema. Amylum starch, where the rhizoids is there, in that part, amylum starch. These questions to expect. Where the vegetative reproduction, amylum starch is present. Chara, Stegleria. Chara, Stegleria is the vegetative reproduction. Bulbils, spherical oval shape, develop on the rhizoid side. When these bulbils detach the parent plant to form the new Chara plant. Chara, aspora. Examples too very important. You can by-head it. Example for the vegetative reproduction in Chara. Another very important, amylum starch. Many times to ask the vegetative amylum starch vegetative reproductive bodies to present Chara, Stegleria, and some species. With these amylum starch, the main node part axis develop multicellular star-shaped aggregations. These star-shaped aggregations full of amylum starch. That's where this name is called amylum starch. And the amylum starch do detachment of plant to develop a new Chara plant. See, listen careful. This amylum starch is most important to ask the expected question. Next one, amorphous branches. These one amorphous bulbils, a group mainly cell irregular shape, lower node and main axis. That is a Chara delicatula, Chara rhizoides, Chara batica, Chara fragifera. These are the example for the amorphous bulbils. Same when bulbils to detach the favorable condition to form to the new Chara plant. Next, coming to the this one also very important. Protonema, secondary proto. Expected 3 question. First question is the amylum starch. This one, amylum starch, or sometimes amorphous bulbils. Secondary protonema, the vegetative reproduction in secondary. These are tubular filamentous which develop from primary protonema, or the basal cells are rhizoid. These secondary protonema from Chara plants. You can observe the secondary protonemas. These are the vegetative reproduction. Next, coming to the reproductive, vegetative you can accept amylum starch, and the second one is the this one, secondary protonema. Here, sexual reproduction oogamous type. Okay, in Chara, another important character here in the Chara, asexual reproduction is totally absent. Which reproduction is absent? Tell me, students, which reproduction is
Asexual.
asexual is
Reproduction.
totally absent. Again, here to expect the question, Chara sex organ's name is different. Male sex organ, female sex organ, globule and nucleus. You can by-head it. This one, very important question. Okay, this is the oogamous type of sexual reproduction. Most species are homothallic. Some are the heterothallic species. Chara Geylanica is the homo. Chara welwitschia is the heterothallic. Sex organs arise on the branches of limited growth or primary laterals. The develop here two sex organs main important. One sex organ, male sex organ name is called globule. Female sex organ name is called nucleus. See here in the picture, this part, green color part is there. This, where the node part is there, primary laterals or primary part, that node to produce the sex organs. One green color sex organ is there, no ma? This one is called nucleus or female sex organ. Inside here, reddish color is the sex organ, male sex organ or globule. Always under nucleus, globule is present. Upper part nucleus, under part is the globule. What are the sex organs name? What is the male? What is the male? Huh?
Globule, ma'am.
What is the female?
Nucleus.
Nucleus. See, this is the globule. See, in this one very important globule, I think so. These are questions are coming or not, but the globule is most important. Okay, this is the antheridium, spermatangium, or globule. The globule size very large, spherical, red in color, red or yellow in color. The mature globule made up of 8 curved sheet cells. See, this structure you can observe. This is the globule, red color, spherical shape, yellowish color is there. How many shield cells? This question to expected. How many shield cells? See, these are the shield cell. 8. You can count it. 1, 2, 3, 4, 5, 6, 7, 8. How many shield cells are there? 8 shield cells. And 8 monobrial cells. Where monobrial? See, here 1 monobrial. This is the 1, 2, 3, 4. In backside 4 is there. 8 shield cells, 8 manubrial cells, 8 centrally located primary capitulum cells. See, primary capitulum, these 1, 2, 3, 4. These are the primary capitulum. Remaining part, another backside 4 primary capitulum. Total, how many capitulum? 8 primary capitulum cells. Secondary capitulum also same. Secondary capitulum inside. See here. The secondary capitulum cells gives rise to many antheridial filament. Antheridial filament meaning these are antheridium filament. Secondary or many these capitulum cells to produce to the single biflagellated anthrozoid or antheridial filamentous. This part is the pedicel. This is a node part. Pedicel. This is a spherical shape. 8 shield cells. 8 elongated manubrial cells. 8 located primary capitulum and secondary capitulum cells. Each and every secondary capitulum cell gives rise to many antheridial filaments. Each sperm mother cell form a single biflagellate anthrozoite. You can by heard it. Globule is a male reproductive organ. The structure to made up of to the 8 manubrial, 8 shield cell, 8 primary capitulum, 8 secondary capitulum, 48 secondary capitulum cells. Each and every secondary capitulum to produce it to the anthrozoid. Okay, did you understood that one? Next, coming to the nucleus. This is the female reproductive organ. See, large in size to compare. See, this is the globule part. Here, this part is called the nucleus. Large in size, green in color, oval structure, born on the node part. The primary lateral. See, here also, it is just above the globule. See, just above the globule. This is the homothallic species. The mature nucleus is attached to the node by the pedicels. This is a nucleus is surrounded by 5 tube cells. Sorry, few tube cells. See, this is the nucleus structure. The nucleus surrounded by, see this part where the outer layer, my arrow is there. These all the surrounded by the few tube cells are present. The nucleus surrounding to form to the tube cells, tube nuke cells. The tip of the tube cell, see this part, the tip of the tube cell form corona. You know, no coronavirus, where the kiritam like structure is there, no? Same here also, just like kiritam like structure. That's where this part of the tube cell above part is called the corona. The tip of top nucleus and the pedicel tube. This is the pedicel, surrounded tube cell. Upper part is the corona cell. 3 cells is there: pedicel, tube cell, corona cells. Okay. Tubes, the oogonial cell, single large egg or ovum. The nucleus contain large amount of starch, oils, receptive stop. These are the important of the after for the formation of male and female to form to the fertilization to form the zygote. These zygotes, see, corona is there. Sperms, you can observe the this is a nucleus structure. All to the anthrozoids are penetrate and gelatinated with the wall of oogonium. Many these anthrozoids enter into the oogonium, but only one of the sperm to fertilize to form to the egg. That is a diploid zygote. See, this is the here after germination of 4 haploid nucleus, same to form to the development of zygote. Finally, here through 3 lower generations are there. These 3 lower generation, see, this is a oosphere wall, main axis, rhizoidal initials. These are the formation of present. Okay, did you understand today's class also? Yes, if any doubts? Students, if any doubt, in Chara? No doubt? Yeah, okay. Shall I close it?
Hello, ma'am. Ma'am.